Bissan Al-Lazikani. So she's really a pioneer in integrating multidisciplinary data and AI in drug discovery. She's a creator of Cancer.ai, the world's largest cancer drug discovery platform. She has a long career in academic and industrial drug discovery, first in Informatica and then ICR. We were able to convince her to come to Houston back a couple of years ago. So she joined MD Anderson in 2021, where she is professor of genomic medicine, director of therapeutics data science, and a founding member of the Institute for Data Science in Oncology.
What I thought I'd do is really take a view of where we are really in utilizing AI realistically in drug discovery, focusing on drug discovery rather than development, but of course they're part of the same cycle. And maybe share with you some success stories, some promising stories, but also kind of a plea for help really about the areas where we need to get together to make it all work better. So these are my disclosures. Yeah, I'm not sure. I think I decided to put it up as a cathartic thing. We can all feel the pain together, right? So the orange and blue plot at the top is the growth rate in genomic sequences compared to the growth rate of FDA-approved drugs, corrected by about 5 years. So I'm allowing a 5-year thing to go between them. And as you can see, we're growing our knowledge, we're growing our data, but we're not translating that to new drugs. And as the people in this audience know better than others, you know, we tend to fail drug trials later on in later stages, phase 2 and phase 3. And oncology, our own personal area, is the biggest culprit. That's where we fail due to efficacy most of the time and more than any other disease area. And all these failures and these late failures, and this is the important thing, the failing late is really, really problematic, which is why we're kind of developing, and again, we're among friends here. So in 2012, we were lemmings, all converging on the same programs and doing the same drug discovery and everything like that. We've grown up a little bit in 2023 because last month we became sheep, and now we're still herding our pipelines. The fundamental thing is somehow, with all these data we're generating and all of this amazing technology that we've just heard about, we're still really not delivering and not moving forward. Part of the reason for my moving to MD Anderson was I felt it was actually a place where I can sort of take a step back, put the whole thing together in a cycle, and look, where are we delivering in terms of utilizing systematic data-informed decision-making, and where are we still reverting back to our old ways, because that should be the areas where we focus the most. And so I like to think of drug discovery and development as this cycle, starting with the patient and ending with the patient, rather than our normal linear model that we follow. And I started putting on sort of hurdles that I envisage on this pipeline, and kind of ran out of breath after 8 hurdles. There are thousands of them, of course. But thinking about, you know, where we're good at, what we can do, well, you know, we're very good at profiling patients in the clinic, and we're getting increasingly better at collecting lots of annotation and clinical data. But we're still not good at actually taking all of that information, as I showed in the last slide, and translating it through to the next step, which is novel target validation. In the target validation, you know, we talk about targets, just target validation. When you think about all the decisions you need to make, from model selection, assay selection, you know, what mutations do you make, rescues experiment, et cetera, all of those, really, the decisions are still being made the same way they were being made 15 years ago. There's very little difference in that area. And you move along this, and, you know, there are bits where we're doing quite well, like we've just heard about sort of the expanding use of generative AI for drug design, for example. Similarly, in the prediction of ADMER tox, you know, there are areas of brilliance that are happening where we are utilizing the data. But by and large, as we're going through these, we do fall across these hurdles. And part of the reason for coming to MD Anderson was really because what we're doing together with Phil and with our discovery biology platform, with our clinical platform, Moonshot's platform, is we're putting this sort of speed highway into place, where we're trying to really put AI in the middle and also the patient in the middle, where we want to start informing absolutely every one of the many thousands of decisions we take with data and with AI, and speed going around this altogether. And we've heard just before now about, for example, the drug design components or the trial design component, which is promising, but we're still nowhere near where we need to be with that. And we go. And this platform that we're putting together in order to link all of these moving parts is called ADA, Adaptive AI Augmented Drug Discovery and Development, completely contrived to call it after Ada Lovelace. If you know who she is, great. If you don't, come and find me after. But the key thing is the following. Whatever decisions we make have to be patient-centric. There's no point making decisions on cell lines and then following them through for 3 years before we check back on the patient. Data-informed and rapidly adaptive. As our knowledge changes, we need to be able to adapt very quickly. The other goal of doing this is redistribute the risk that is associated with innovation to earlier on. Let's test all of the hypotheses really, really early on, so what we move forward to phase 2 and phase 3 are the ones that are much more likely to succeed. It's a good dream, but hopefully I'll show you how we're starting to make some headway towards it. So target identification. Those of you familiar with our research know that every now and then we publish a paper that goes like this. We've taken a bunch of cancer targets and identified lots of promising druggable targets within those. And every time we do this analysis, we discover that somewhere between 5 and 10% of any list of genes that you find reported in a cancer genomics paper in supplementary table 2, or whatever it is, 5 to 10% of those genes are druggable, are useful, and are ignored. No one's working on them. We've showed this enough times for it to become sort of kind of frustrating a little bit. So why is it that we're ignored, and how can we really bubble them up to the top to make them more interesting for people? And the reason they're ignored is because in cancer genomics, we're still dreaming about these things. You subtract the normal tissue away from the cancer tissue, you end up with a really big spike of a driver gene that happens to be perfectly druggable and easy to design a drug for. You design a drug, enter clinic, it works, everybody lives happily ever after. And in reality, what we do end up with whenever we do these exercises is a big cloud of genes, most of them we've never heard of at all. And we're trying to think which of those are we going to invest, drug discovery, but before anything even, just target validation. Of course, we're going to gravitate to the genes that we're more familiar with and the pathways that we're more familiar with. And that's a large part that's driving these lemmings and sheep and whatever it is we become in 10 years' time. Hopefully none of those. So what we're trying to do is really figure out how you utilize data in order to help us really isolate the signal even from a large amount of noise. A few years ago, we demonstrated that actually, well, we have many things at our disposal, but one of the things I'll talk about is a few years ago, we've demonstrated that the molecular networks, the molecular interaction networks, if you forgot that they're protein networks and started treating them as just communication networks or like social networks and started deriving patterns from those, what we discovered a few years ago was that actually the communication patterns in molecular networks are very, very good at predicting cancer drug targets. Turns out that cancer drug targets lead very different social lives to your average protein, and also to drug targets from other therapeutic areas. And that's driven a lot of our work since then to try and understand how we can utilize this different cellular communication, these different social lives of proteins in order to help identify novel targets. So initially, a couple of years ago, we worked with the International Cancer Genome Consortium, of which I am part, for prostate cancer, in order to try and start constructing what we think these signaling networks look like for prostate cancer. And that was a really simplistic way of looking at it, but what we identified is if you take the genomes of prostate cancer patients, do the normal bioinformatics and identify, supplementary table 3 with 73 genes in it, what you can do is you can start find clustering of communication between these 73 that point you to certain biological processes that are happening, But importantly, what it enables you to do is bring in other proteins that are really essential for the communication, but you would have missed because they're not mutated or they're not very frequently mutated. And so basically, here's what we do. We take the mutated genes or whatever it is. This applies to any kind of molecular measurements. Could be RNA-seq, could be proteomics, could be anything. We put them all together. We get the seed list of genes here. And then what we do is we have a big reference background interactome. The first thing we do is we construct the links between these seed genes, and then any fragments that we have, we connect them using a specific algorithm using this interactome. So you end up with a big network that contains both mutated or altered proteins, but also other proteins that are deemed by the algorithm to be important for the communication. Once we do that, we apply a probabilistic model to really test how necessary each node is to the communication. And so this protein is mutated and is really important for the communication. This protein is not mutated, but also pretty important for the communication. So what we then do is use this probabilistic model and rank the genes based on what we think are the most likely molecular vulnerabilities for that disease. So that's how the algorithm works. We've applied it to all of the solid tumors in the TCGA as a test set. This is the lung adenocarcinoma study. And basically, we do this, and then we get a ranked list of genes, which you see here. And those of you familiar with lung cancer, you'll recognize many of the usual culprits in there. If we look at the top predicted genes from our model and overlap them with the top mutated or altered genes, which is typically the way people go for these in terms of just abundance or frequency, we find there's actually very little overlap between them. And that's partly because our model is still a work in progress, but partly because a lot of these heavily mutated genes aren't important. They're just incidental. Or altered genes, they're just incidental. So we were very happy with the fact that we were able to find a lot of the culprits. And I would note things like, for example, EGFR ranks second in the list after P53, although it's only mutated in 12% of this cohort. And you'll see beneath it, there are other genes that are mutated in much larger fractions of the cohort that do not score as well in our model as EGFR. So we thought, well, that's very promising. So how good is our model at actually identifying these molecular vulnerabilities if they're not mutated at all? We know from prostate cancer, because we showed it before, that it can identify the androgen receptor as a key player, even if it's not mutated, which it rarely ever is in the patients. So what we did was we did two exercises. We set EGFR to be wild type in our initial analysis. We pretended it was not ever mutated or altered in any way, and then rebuilt the model and reassessed it. And even if you set EGFR to be wild type, it still comes up as the second most important protein in that network. So it's telling you no matter what, EGFR is essential for that communication. Similarly, we set KRAS to be wild type, and this time KRAS dropped from 4th to 8th, but it's still in the top 10%, which for 10 genes. For us, it's really great, because if you know 10 or 20 genes that you want to take to your first stage of target validation, well, that's brilliant, that's great. So we're really encouraged about this. We then took all of these solid tumors and decided to do the more global analysis and asked, if you take the top 20 genes that are regarded important by our probabilistic model versus the top 20 genes that you would get from just the ranked mutations, how do they perform when you look for dependencies, genetic dependencies in DepMap, or drug sensitivity using the Sanger panel? And these are the solid tumors that we've tested. I think there's 15 of them that we've done the analysis for. The colored bars are the genes among the top 20 from our model, from the predictive model. And this is how many of them are actually genetic dependencies identified in DepMap versus what you see using just the rank list, corrected rank list based on sort of abundance or frequency in patients. And this is just the distributions here, so you can see them side by side with each other. So definitely, this model that is trying to look for where the communication really is happening is doing better in general at identifying true dependencies than just the traditional way we typically go down lists and identify targets. And this on this side, where we took each of these genes and looked up its drugs that have been tested in the Sanger panel for drug sensitivity. The green is the genes from the model. The brown is the genes from if you just rank them by frequency. And again, you see that the probabilistic communication model is massively outperforming what you would have done otherwise. So nowhere perfect, but how good is it to be able to say that actually if you've got 20 genes, the chances are the most important ones are already in there. So you can start triaging and informing your validation experiments in a much more objective and rational way. So of course, we live in the real messy world, and in MD Anderson, we're particularly interested in rare and complicated cancers. This is work we've done with uveal melanoma, where we've taken sort of about 150 patients, which is an immense number for uveal melanoma, and we've done molecular profiling and done essentially unsupervised clustering of the patients, and the patients cluster molecularly into these 3 major clusters. And one of them is very heavily representing the metastatic subcohort. And what we can do now is not only have a network, a communication network for the whole cohort, you can actually start breaking it down into subcohorts and sub-subcohorts all the way down to an individual patient. And what we have here is, you'll forgive me because we're working on the targets at the moment. What we have here is the molecular communication network for the metastatic subcohort. Each block you see is a gene, and the darkness of the color represents how highly ranked it is by our probabilistic model. The size of the block represents how many patients it's mutated or altered in some other way in. And you see, dark ones that are, ranked highly by the model and also mutated in a larger number of patients. But you also see some other dark ones in the middle, which are not altered or not mutated at all. We are currently validating a lot of these. So I'm going to show you just one example of here. This one happens to have a chemical tool available for it. Chemical tool available for it, and here are the initial data that we're finding coming out of our experiments. So we are showing response to this particular target. What the model also tells you is it starts giving you hints for it. It thinks that the place of activity is happening. Is it sort of more centrally, more cancer cell intrinsic, or is it happening in the microenvironment? So for example, that particular gene, the big dark triangle, is actually coming from the microenvironment, and we're working with our digital pathology team in order to try and identify between looking at the patient pathology maps and single cell profiling to identify the key culprits and how we can target this gene in patients. Now, I talked about informing target validation. So hopefully I've showed you how we're working to inform target selection. There's a whole piece about PD biomarkers as well that I don't have time to discuss today. But one thing I wanted to bring up, because this is a real thing that we're facing, is doing the easy experiment versus doing the right experiment. And so we're lucky enough to have quite a large number of cell lines available to us to do the validation in. The triangles, this is the same plot I showed you before. The triangles are cell lines, and they're clustered with our patients. And you'll see some of them are closer to the patients than others. They tend to kind of fall roughly in the same regions as the patients. That's quite encouraging, because you could say that, this gray cell line is probably more representative of that cluster than the other cell lines. But now, I'm going to ask you if you can point me to where the easy to work with cell line is. And it's this one. It's the one that looks nothing like any of the patients. And unsurprisingly, that's the one that is the workhorse that everybody is publishing on. And so what we're working with the discovery biology team now is really to make the models that work but represent the patients, as opposed to build too much sort of sandcastles on these models that just behave. How much time do I have? One minute 2 minutes. Okay, I can do this really quickly. Just wanted to, I know some of you know CANSAR, some of you don't. Hopefully the ones of you that know CANSAR, some of you would be fans. So CANSAR has now moved to MD Anderson, is being supported moving forward. A lot of the work that we do, the algorithms that we develop, we actually do put them publicly and freely available through CANSAR.ai. And so please use it and give us feedback if it's good or bad or ugly, whatever you like. And we look forward to work with the community on that. One of the things CANSAR is kind of sort of known for is the assessment of drugability. And we're expanding the drugability to well beyond sort of the availability of small molecule binding sites to looking more generally at the behavior of the chemistry, looking at neodegrons, looking at the network, for example, but also accessibility to biotherapeutics, et cetera. And with that, when you put all of these together, we believe that over half the human proteome has the right access point and can be drugged. You just need to know what modality and what site to go after. And this is something that we're utilizing AI constantly in order to inform. And it wouldn't be a drug discovery talk without mention of AlphaFold. So I just thought I'd show you sort of our analysis of AlphaFold. So AlphaFold has been fully analyzed. It's all available in CANSAR. If you're interested, you can look for all the druggable pockets in CANSAR. Of all the cavities we detected using AlphaFold, 6,500 of them were druggable. Almost all of those, if you said, well, I'm demanding that the cavity is consisting of high confidence amino acids in terms of the prediction, almost all of them pass. But if you say 70% of them have to pass that, again, almost all of them pass, just under 6,000 cavities, which is great. The 6,000 cavities are almost one-to-one, and they correspond to 5,800 druggable proteins. When you overlap the druggable proteins that we identify from AlphaFold versus what's druggable in the PDB, we find that there is an overlap, but it's not as big as you think there would be. And looking at the reasons, for example, AlphaFold does really, really well with membrane proteins that obviously are deficient in the PDB, so that makes sense. But actually, when you look at this set, they fall into two major categories. One of them where actually, it's just that snapshot that was published by AlphaFold doesn't have the structure formed correctly for the druggable thing. But if you ran AlphaFold and took snapshots, 10 snapshots, or ran, MD or Monte Carlo, then you would rescue quite a lot of them. But other ones actually have the structure so wrong in the region of the druggable cavity, it is simply irredeemable. And another thing that we and others have found is there aren't that many novel folds that are found by AlphaFold. And that's either true, because structural biologists have managed to solve all the possible known structural folds, or it's not true. And AlphaFold is still stuck, basically, in what we know. And I think it's more likely to be the latter. So just want to summarize by saying that we really are ready for big time in AI. We just need to work together to start addressing the tricky questions as opposed to just show our cleverness in the easy questions. We are lost in the amounts of data, but also there are data that we really need to bring together, and we need to develop the small n, the small number of patients and big measurements. But I'm really excited about the next 10 years in drug discovery, and I'll thank my collaborators and my amazing team, some of whom are here today, and funders, and most importantly, the patients.